Okay, it's time to get going. Just 2 more classes. So the whole idea here for this class, we're going to finish chapter 11, and that will be it for the material for the final. So then we'll just start working on final review. So I just wanted to show you the resources that I posted earlier this week about the final. So I put it at the top of the lecture module because I think it's super important. Basically, it's just like the midterm resources that I gave to you previously. It's just, cumulative for the final. So here you will find the final makeup form. Final midterm, okay, so that's a typo. So if you do have a conflict with the final exam time, which is Thursday at 2 p.m., probably in this, I'm assuming it's in this room. It's a different form, all right? Different from the midterm makeup. So this one has its own dedicated form, just so you know. I have the learning objectives from all of the chapters. So this final is cumulative chapters 1 through 11. Here's the scratch paper that you'll get. It's the exact same one from midterm 3. FYI, I'll ask you guys to please test your Respondus lockdown browser just one more time, so I have that linked there, so you can test it before you come into the room. And then I have all the materials you have to study from. I have it broken up into new material, meaning I, put these things together fresh. Most of them are questions you haven't seen before, or at least with new numbers. And then here are all the older study materials, so back midterms, recitations, overview sheets. All right, so they're all there, all of them. Do you ever write a word so many times that it doesn't look real anymore? Yeah, that's what I think about the word key, all right? I just wrote the word key over and over, and I thought it wasn't a word anymore. I had to check dictionary.com to make sure that it was still right. All right, so you've seen all the older study material for before, but the new study material is new. Here is it's not a practice final, all right? I'm not allowed to write practice finals, but I can instead just give you 100 questions that cover all the chapters we went through, and you have that to choose from, to study from. I just can't put together a practice final. I'm not allowed to. But yeah, there's like almost 100 questions here for you to go through, and the key is there. So a problem set for you. And then I have the Zoom in-class review packet, so we'll start that today. So if you're looking at this page, just go ahead and download that now. And we will complete the rest, today, on Thursday, and then we'll do anything left over during the Zoom session next week. And I have that linked here, right, for the Zoom session next Tuesday. So that's reading day at 6 p.m. to 8 p.m. I will record it. It will show up on that page, just like always. All right. Any questions about the final stuff? Okay. And then the last idea here, I sent an announcement about the last homework due dates. All right, so homework 10 was due yesterday, Monday. I extended it a day because Pearson went down for a couple hours, so I gave you an extra day for homework set 10. But the last homework set for nucleic acids is due on Monday, so the very last day of classes at midnight. So I gave you an extra day there. It's a really short homework set. But an idea I want to remind you about the late homework policy. So any question that you haven't gotten to, you can still do for partial credit for up to 10% of the original point value. So if you just, if you did 30 out of the 32 questions in a homework set, you can revisit those last 2 to get extra points. It's not going to affect what you've already submitted. You'll just get extra points in that regard. So it's always advantageous to just do as many problems as possible to get those points, and joke's on you, you're learning at the same time and getting more prepared for the final. So do revisit those late problems or late homework sets. You can still complete them for partial credit. The last day that I will allow late homework to be turned in is Tuesday at 8 a.m. All right, I need to submit and sync the grade book with Pearson one last time, and I plan to do that on Tuesday morning. So I have the deadline for late homework Tuesday at 8 a.m. So that's the last, basically whatever your grade book in Pearson says at that moment is the homework grade that you will get that will be synced into the grade book for calculating your final grade. All right, any questions about that? So Tuesday, 8 a.m., that's the last that late homework can be submitted. So you can still revisit like things from chapter 1 if you didn't get to them. You can grab some more points on your way out, right? There's that. All right, so let's finish up nucleic acids. Yeah, all right. So nucleic acids. We left off talking about the components of nucleic. Oh yeah, question. The grades. I don't know. There is a day where the lab supervisor says to all the TAs like, hey, you need grades in by this time so we can calculate it for the grade book. Same thing with recitation. I had to like yell at my TAs like, put those grades in. I anticipate it will be before reading day, because you guys don't have like labs anymore, do you? Yeah, you're done. So this is the time where all the TAs are crunching their books, and hopefully those final grades should be in soon. Is it Tuesday? Sorry, Aaron's talking to me. Aaron, what was that? What day was that? Aaron, can you say it again? Okay, so Aaron just said that the last day for lab grades for the TAs is Monday the 25th. Okay, so before reading day, all right? So, and that's, the deadline for TAs, so you might get it before then, yeah. It's already posted. I just posted it yesterday, I think. Did you see that? Yeah. Oh, I guess I should. Do you get an announcement or like okay, all right, yeah. Midterm 3 is posted, all right? I sent the paper ones to people. Sorry, I should have made an announcement. It was last night, so don't worry, you didn't lose out on time. Yeah, so midterm 3, the grades and the results or your responses are posted. I'm not gonna write a key because I'm sick of writing keys. Okay, so any other questions about the end of the term? Yeah. Oh, so the late policy, so, the due date passes, right? The due date passes, and so any questions that you complete before the due date, you can get full credit for them. And so most of the homework sets are worth about 50 points each, right? But let's say you only did half of the homework set. So everything you did went to the grade book and you earned the maximum amount of points or whatever for what you did. But those last questions, you can still do them up to 10% of their original, the original points that they were worth. So any late questions, it's on an individual basis. So every individual question you can still submit for a late penalty, but it's not going to affect what you've already submitted. Does that make sense? So doing late questions that you didn't do will only increase your grade. It'll never decrease your grade. Does that make sense? Okay. And basically the late policy is there's a 2-day grace period after a homework set is due, but for the majority of any questions you haven't done so far, it's past 2 days. So the maximum you can earn is 10% of the original point value. Yeah. Didn't do. I don't think I don't know. If you still have attempts left, I don't know. I don't know that detail. If you missed it and never finished it, I think you can still do it as long as you still have attempts left. Does that make sense? Yeah, because you didn't earn anything yet, right? And so as long as you have attempts left and you didn't hit I give up, then you should still be able to complete it and get a couple extra points, okay? Other things, okay? All right, let's get started. Nucleic acids, DNA, RNA, stuff like that. All right, so we talked about the different components of nucleic acids. So we talked about the nitrogenous bases. So there's 3 components. There's a nitrogenous base, a sugar, a ribose sugar, and a phosphate group. That's how we describe a nucleotide, which is basically the unit within a whole nucleic acid strand. So hopefully these terms kind of sound familiar. You know, we've been talking about amino acids and building them up to peptides. It's the same idea here, just that nucleotides, they have 3 components, and then we combine multiple multiple nucleotides to build a nucleic acid strand, which we call DNA or RNA, based upon what kind of ribose sugar that we see. All right, so nucleotide has those 3 components, while a nucleoside actually only has 2 components, so it doesn't have the phosphate group. Right, so that's the only difference there. So all of these that you see right here, these are nucleotides because they have the phosphate group. You see the sugar, you see the nitrogenous base, and so the nitrogenous base is kind of what gives the bulk of the name for these nucleotides. And basically the name tells you exactly everything that is in it. So adenosine monophosphate contains the adenine base plus the sugar. So in this case, because it doesn't say deoxy, it's just the regular ribose sugar. So there's an OH on carbon number 2. We talked about that on a previous, I'll just quickly show that. Right, where's my sugar? Here. So our ribose sugars. So what you want to focus on to determine whether a sugar is ribose or deoxyribose is you look at carbon number 2 and you just see, is there an oxygen attached to that carbon number 2. If there is, then it's regular ribose. If there is no OH group oxygen attached to carbon number 2, then it's deoxyribose. So no oxy on the carbon number 2. All right, and so that's where we get the names of RNA and DNA. Ribose nucleic acid has the ribose sugar as its component. Deoxyribose is found in DNA. So that's the big difference there. So you see all of the nucleotides found in DNA have that deoxy prefix on those names because we use the deoxy sugar, the deoxyribose in DNA versus in RNA, we use the regular ribose sugar. So a question that you'll probably run across if I give you the name like this, like DAMP, DAMP, I could ask you something like, where would you find DAMP? And the big idea here is there's a D, meaning deoxy, in front of the name, so it has to be found in DNA versus in RNA. RNA, you would only find AMP, not with the D. All right, and then a little kind of detail here is that in RNA, we have uracil as one of our nitrogenous bases, and that is analogous to thymine. That we find in DNA, so you will not see U's in DNA, you will not see T's in RNA. So that's kind of like that last detail there. So you would never see TMP. TMP would not exist in our bodies without the D because thymine would not have a ribose sugar attached to it because that's with RNA, and kind of vice versa, you would not find DUMP in RNA because deoxy is associated with DNA, all right? So those are those big ideas, all right? And the names tell you exactly what's in it. Monophosphate tells you there's a phosphate attached to it, so that has to be a nucleotide within there. All right, so those are all of our reading and naming our nucleotides. So what's next here? How do we actually form the nucleic acid? So nucleotide is our building block. That's like analogous to amino acid or monosaccharide, like a glucose unit. Now let's put them together to give us a strand that builds our DNA or RNA, whatever. So hopefully you see all of the relationships and kind of repeating themes here. We undergo a condensation reaction to connect multiple nucleotides together to build a whole nucleic acid strand. So the big idea here, we're building our nucleic acids from units, our nucleotide units. The type of bond that is made is called a phosphodiester bond. Kind of like when we talked about building amino acids, we made peptide bonds. When we built sugars, we made glycosidic bonds. So here, when we talk about nucleic acids, we are using phosphodiester bonds, but really it's just a condensation reaction. But what we're looking at here, the bond being formed when we combine these two separate nucleotides, we're forming a bond off of the phosphate group onto the hydrogen, the OH group off of carbon number 3. And so since we're making a kind of like a phosphorus oxygen carbon bond, that's why we call it a phosphodiester, that there's a phosphorus group here, there's an oxygen and then a carbon off one side, and then an oxygen and a carbon on the other side. And so this is why we call it phosphodiester. This is the bond that we're talking about here. But really, it's just a fancy condensation reaction. You can track the two hydrogens and the oxygen being lost to connect these two building block units. So in this case, phosphodiester bond is analogous to a peptide bond, right? When we talked about amino acids, and that is analogous to a glycosidic bond when we talked about sugars. So reoccurring themes. All right, so we put a bunch of these nucleotides together. We form our nucleic acids. So what you see here is we are building a backbone, just like when we talked about our peptide backbone. Is that what's next? Yes. So we are connecting the phosphate group to a sugar. And then when we connect a bunch of nucleotides, we're just making phosphate sugar over and over and over again. All right, so the question is, okay, well then where do the bases come into play? They're actually dangling off of that phosphate sugar backbone. All right, doesn't that sound familiar? That sounds familiar, right? Familiar to amino acids when we talk about the dangling R groups. Same idea here. So just with nucleic acids, we have a phosphorus sugar backbone with those nitrogenous bases kind of dangling off. And now you can see, oh, we can put two nucleic acid strands together to get us our double helix of DNA that we know and love. And so the idea here, once we have this, we connect a bunch of nucleotides. The order of our nitrogenous bases, we typically identify nucleotides by what kind of base, is it A, G, C, etc., by what nitrogenous base it is. The order here really matters, just like when we talked about peptides. So the order matters here. This is what we call the primary structure of a nucleic acid. Same idea as when we talked about peptides as well. The order of the actual nitrogenous bases is the primary structure. All right, so right here, that order, so this is GTCA. The order of those nitrogenous bases is our primary structure. It is, again, important, the order, so we need to make sure that we read it in the correct way. So typically how we read nucleic acids is we always start from the free 5-carbon end off of the phosphate sugar backbone and read towards the 3-carbon end. And so that hopefully sounds analogous to when we did amino acids, and we typically read from the N terminus to the C terminus. So it's the same idea, but this time we're reading 5 to 3. I'll show you where that 5 is. Like if I go back up here. So here we have two nucleotides together in just like a 2 nucleotide strand. So here, this is our first nitrogenous base. This is what we call the free 5-carbon end, because the 5-carbon is the tail. It's the tail. So that's the one that's free. So when we're reading left to right, we typically start with the nitrogenous base that has this free 5-carbon versus we're reading towards the free 3 end over here. Because see how the 5-carbon is attached to something? It's attached to something there, so that's why we don't start on this side. So we're reading in this direction. Go to 5 to 3, just like in amino acids where we did N terminus to C terminus. So the order here really matters, all right? Bases hanging off the phosphate sugar backbone. It looks just like a peptide. R groups are just different. They're nitrogenous bases instead. All right? So that's how we form a nucleic acid. So how does this all kind of tie together and build DNA? So DNA is the like instruction manual for our bodies to make proteins and build us and make us grow strong. That's why we eat our vitamins. So we're forming nucleic acids. Now let's build DNA. Nucleic acid, the order of those nucleotides tells us our primary structure. Now when we think about DNA and its double helix, this is now us getting into the secondary structure of DNA. So the story about DNA and how we kind of, this is a little side story about how we kind of got to the idea of scientists discovering what's the purpose of, or what's the composition, what's the structure of DNA. It involved a lot of different people probably fighting and having controversy over, what is the true structure of DNA. So here's a little bit of data that allowed scientists to kind of glean and understand a little bit more about the purpose of the nitrogenous bases. So this experiment by Erwin Chargaff determined the composition of DNA, the different components of those nitrogenous bases. So we have A and T, C, and G. Those are our nitrogenous bases. So this guy was able to get the percent composition of each of those nitrogenous bases of different DNA strands in different mammals and different species here. So put yourself in their shoes and pretend like you know nothing about DNA, that you never took biology, and you don't know that A goes with T and G goes with C. They knew nothing. All right, so all that these people had to kind of understand and come up with conclusions were data that they actually measured. So within this data, Chargaff was able to determine the composition of the different nitrogenous bases in E. coli. And so what he found is that there was basically an equal amount of A, T, C, and G within E. coli. That doesn't tell us that much to me. Like, it's just like, oh, they're all equally like represented. All right, so to me that doesn't mean that much. But then once we start going down to other species where he was able to determine the composition of those nitrogenous bases in the DNA. So for yeast, he saw that, yeast is about 32% adenine and 32% T. So he saw that, those two matched. And then C and G also matched their percentages here. And then as he went down to other species, he saw that A and T always matched and C and G always matched. And all four of those components always add up to 100. So he was able to kind of think here, if we think about the structure of A, G, T, and C, remember A and G are purines. Those are the large rings. And the pyrimidines are the smaller rings. And so what he was able to say, so we got A and G are purines here. T and C are pyrimidines. So these two. What he saw is that the number of purines always equal, or I guess the percent of purine was always the same percentage as the pyrimidine structure here. All right, it was always 50-50. And then from that, he was able to see, a pyrimidine always goes with a purine. That's what we have here, that A and T go together. So A and T go together, and G and C go together. So that's what we were able to, did I do this right? Sorry, I think these two go together, these two go together. Wait, no, sorry. A and G go together. T and C go together. So those are pyrimidine, and these are purines here. And so from this data, he was able to get that the actual relationship that we use today in biology to figure out the nucleic acid order. And it all came from numbers, but we had to do it with multiple different species. And then we saw that this happened in any species. So if we only looked at E. coli, we might never have actually gotten to the point to understand, A goes with T, G goes with C. All right? Okay, so let's keep going. So that kind of tells us how we get the order of DNA and how those two strands always go together. So the story about how we actually got to the structure of DNA involves a lot of different people. So you see that right here. So Lawrence Braggs, he was responsible for X-ray crystallography, and that's how we were able to get the structure of DNA and the double helix structure of DNA. So we essentially X-ray crystallography, we essentially bombarded DNA with X-rays, so light, and we kind of saw how does DNA diffract that light, and from the light that's collected, we're able to kind of back calculate what does the thing look like that made that diffraction. That's essentially how X-ray crystallography works. So the next person here, Linus Pauling, he is well known for a lot of physics and fundamental chemistry, but he was actually the one to kind of determine the alpha and beta secondary structures within amino acids. So that kind of gave paved the way for the ideas of, oh, DNA can have structure as well. So he did alpha helix and beta pleated sheets. All right, Erwin Chargaff, we just saw him. He got the whole A equals T, G equals C thing going on there. All right, and he was basically credited with both of these to get the whole idea of the double helix and how the double helix of nucleic acids or of DNA here. So together with this idea of, okay, the purines and the pyrimidines go together when forming your double helix, what the actual structure got to. Put all of those ideas together to get the structure of DNA. Because really the ordering of the A, T, G, Cs, all of that, that tells us like the composition of our nucleic acid, of our DNA strand, but these guys were able to put it together to get the actual structure. And this paved the way to us actually doing, or chemists, biologists doing crystallography on a DNA sample. And so Rosalind Franklin was the one to actually do the X-ray crystallography of DNA. So she was able to do a bunch of things. She was working with DNA, all right, was able to purify it enough to get the density of DNA. So this was kind of her project, and she was the one to actually do the X-ray crystallography on a certain DNA strand. Do I have it here? Which one did she do? She took the clear picture. So this is what the X-ray crystallography, the diffraction pattern looks like of DNA. She was the one that took it. She also determined the density of DNA as well. But her boss then took this picture and gave it to these guys, all right? So she took the picture, but Watson and Crick were the one to actually interpret the picture and what it means. Because to me, like, pretend you don't know that DNA is a double helix. And you just look at that picture. I don't know, it doesn't look like anything. It's just like this weird looking X type of thing. But remember, this picture is the diffraction pattern of what DNA makes when you hit it with X-rays or light. Like if you think about, do you all still use CDs? Do you know what a CD is? Okay, all right, thank you. So if you have a CD and you like reflect like sunlight off of it, you see like a cool pattern come off. Pretend you didn't know what a CD was and all you could see was the pattern it made. So basically Watson and Crick took the picture, the light that was being diffracted, and back calculated, oh, this is what the CD must look like, or this is what the thing that made the diffraction must look like. So that's why their contributions were Nobel Prize worthy. Unfortunately, Rosalind Franklin died before the Nobel Prize was given out to these guys. So I think it was like 1962. I think the whole structure of DNA, that whole Nobel Prize, I think it went to like 4 different people. I have it on the next slide. It went to a bunch of people, but unfortunately, Rosalind Franklin died before the Nobel Prize for DNA was given out. So who knows if she would have gotten it or not. I'm not sure. I'd like to be hopeful and think yes. And unfortunately, she died at a pretty young age, and probably the reason was because she was constantly taking X-ray crystallography, so she was constantly working with X-rays, and this was before the time that like personal protective equipment was well understood at the time, so probably her work was killing her, unfortunately. But that's why PPE and safety is very important now. So if you work with X-rays in the hospital settings, you're fine because we have proper precautions to keep you safe. Now, all right, so here, so that's Watson and Crick, all right? That's where they interpreted this pattern and basically came up, oh, this is the double helix structure of what DNA must look like. So I guess those guys like recreated their picture. Look, see, but they're like super old. I found this picture. I thought it was funny because they're just like super old. So they recreated the picture of how they determined what the double helix looked like. We're taking two nucleic acid strands and twisting them together, where the nitrogenous bases are hydrogen bonding to one another. That's why we get A equals T, G equals C. Those are the nitrogenous base interactions, and they're twisting together. So here are all of them actually receiving their Nobel Prize. I think two of these people are Watson and Crick, and one of these other guys is Wilkins. I don't know who. I have it here, but it's a blurry picture. All right, so hopefully, I like to think if Rosalind was around, she would be in that picture too. So I will be hopeful. All right, so that's the story of DNA and how we got to this point. That was a huge undertaking, all right? Like, I feel like we kind of take for granted when we learn some of these things, like A goes with T, G goes with C. It took an army of people to determine that, all right? And it took years and years of research to just figure that out, and you know, it just gets like a page in the textbook that we read about now. But that's basically how chemical and scientific progress goes. It can be very, very slow. And it builds off of one another. So it was very slow back then, but now you're learning it now, and it's kind of like an afterthought, like, oh yeah, of course that those two will associate that A goes with T because the hydrogen bonding fits together. It's so easy now to look at it, but it took a bunch of people and a Nobel Prize to realize that. All right, so I think that's one reason I love science. Okay, all right, so that's DNA. So what is like the purpose of the whole idea of the double helix? We have two nucleic acid strands connected together and twisted together. What is that purpose? It allows DNA within our cells to replicate with one another. That since we have two strands and they're complementary to one another, if they unzip and new nucleic acids come in and make the pairing, you can get two new DNA strands out. So I like to think of it as it's like you ever like get a instruction manual for something and it comes in English and Spanish. Okay, so you have like a double copy, right? Like you have two instruction manuals there, but just in different languages. So if you had two people, you could from one manual build two different things. Or the same thing because you have the instructions in two different languages. So it's the same idea here. When you have a double strand, you have copies that are complementary to one another. So if they unzip and you make copies, photocopies of those, you can get two out. And that's the whole idea of DNA replication. That here is our original parent molecule, that these two strands unzip, and this purple is the photocopy that's made. So from one parent, we get what's called two daughters within here. And then this can happen like an infinite number of times to get more generations and identical looking DNA strands. And then this is kind of more of a, here it's like a very comical representation of what's happening, but when your DNA strand unzips and you make a photocopy, when I say photocopy, I'm saying that the corresponding nucleotides are literally coming in and making the correct pairing here. So you're seeing this original double helix unzip, and new nucleic acids or nucleotides are coming in and making those base pairings. So that's kind of what's happening on the particulate level here. And then the question is, well, how does DNA know that A goes with T? That whole idea, or like how does DNA know that G goes with C? and it has to do with the hydrogen bonding, what the molecules, those nitrogenous bases actually look like. And because those hydrogen bonding interactions match, you have a very, very strong interaction. So that's why A only goes with T. Because of the hydrogen bonding matching here, all right? But sometimes, you know, DNA can get it wrong and there can be mutations. We're not gonna talk about mutations in this class, but the whole idea is, okay, yes, this pairing can happen, but we can get it wrong sometimes, and bad things can happen if we get it wrong. Sometimes bad things, sometimes nothing happens. But yeah, sometimes bad things can happen when mutations and basically those base pairings are wrong. Okay, so that's DNA replication. That's how one DNA strand makes two daughter DNA strands. We will talk about RNA in a little bit and the purpose of that. So secondary structure of DNA. We talked about primary structure, right? Before, primary structure is just the order of the bases. Okay, so like right here, when you look at one of the strands, we start at the 5 end and it goes A, C, A, you know, etc. That is primary structure, just the order of the bases. Secondary structure in DNA is the formation of the double helix, where we have two complementary nucleic acid strands that are hydrogen bonded together via those nitrogenous bases. So secondary structure is the complementary base pairing, the whole idea that A goes with T and G goes with C, that we need that complementary bases to twist and make our double helix. All right, so the detail here that I want you to get at is as long as we have one of the orderings of one of the strands, we can get the ordering of the other strand, right? We only need to know one of them, and we can get the other strand because we know the complementary base pairing, that A goes with T, G goes with C. But the detail about how the nucleic acid strands actually align their hydrogen bonding is that the two strands are what we call anti-periplanar, meaning that if we always read the strands 5 to 3, right? Like when we read like 5, the free 5 carbon side, and then you go like A, T, G, C, whatever, and then you read on to 3. If we look at the other strand, the complementary strand, the 3 side starts with the 5 side. Does that kind of make sense? So if these are our two strands, right, so another way to draw this would be like, okay, we have, you know, those nitrogenous bases, this is the 5 side, here's the 3 side. If we were to unzip that and look at the complementary side, the complementary strand, this one starts at the 3 side, this one starts at the 5 side. That's what I mean by anti-periplanar, that all these double helixes, when we are making photocopies, they have to be kind of, they start oppositely. One of them, I like to think of it as like the template, goes 5 to 3, and the newly formed one is 3 to 5. Okay? All right, that's important because sometimes when we're trying to predict RNA strands, knowing whether you have to start at 3 or 5 is important. But as long as you're given your DNA template and which side it's starting on, you'll be fine as long as you know that it's kind of opposite here. When you build one, you build it backwards within there. All right, so secondary structure is the double helix, the base pairing. So that's kind of similar to the secondary structure of peptides and amino acids, where we had helixes and beta-pleated sheets and all that. Where DNA differs from our idea of peptides is the tertiary structure. So we said tertiary structure, I guess it doesn't differ that much. Tertiary structure, we said in amino acids, was the folding of proteins. In DNA, it's actually the supercoiling of making of chromosomes within our cells, which I think is pretty cool. You know, I've shown you what DNA looks like, but when it's actually in our bodies and compact, it's in the form of a chromosome, where we take all of those nucleic acid strands, your DNA, we wrap them around what's called a histone. It's just a special type of protein, and it packs itself into this X-shaped chromosome. I think that's pretty cool that our bodies do this. And so those DNA pieces that are within our bodies separated into 46 different pieces, and we have those chromosomes there. So it's cool. So here's like, you know, your strand, Each of those strands are wrapping around these histones, and then that is supercoiling into itself, kind of making this long rope. That rope then forms itself into a chromosome. All right, so there's a lot of different structural order within there. But as long as you know that tertiary structure of DNA is a chromosome and it uses supercoiling to get it to be more compact so it can fit in your cells, that's basically all you need to know there. All right, so there's our chromosome, and it's made up of DNA. Okay, right, so that's DNA. That is the ordering, the primary structure of DNA, the ordering of those nitrogenous bases gives us the instruction manual, gives cells instruction manuals to make everything in our bodies. If you think about our bodies as being a big IKEA warehouse or something, all right, that those chromosomes and your DNA contain all of the different instruction manuals of all the different pieces and stuff. Have you ever built something from IKEA? Yeah, okay. Our bodies, every single one of our cells contains the instruction manuals for all of those different pieces of furniture and things that we have to build within our bodies. So how we actually do that, how we actually do the building is through RNA. So DNA holds the material, holds those, it's holding the PDFs for those instruction manuals. RNA is the one that actually does the building. Does that make sense? Okay, all right, so let's do it. And yeah, we're not going to talk about these things, so we're almost done here. All right, so just a quick comparison of RNA and DNA. We already talked about it a little bit in terms of naming. R in RNA stands for ribose. D in DNA stands for deoxyribose. So that's like the big thing, that the ribose sugars differ. Is that the first one? Yeah, the sugars differ. Another difference is the bases. We already talked about that. It's U instead of T for RNA. And do note that both of those go with A. All right, so both of RNA and DNA use A. All right, but it's the complementary nitrogenous base is different for RNA and DNA. U is for RNA, T is for DNA. All right, RNA is a single strand of nucleic acids or nucleotides. It's a single strand. It's just one. Versus DNA is a double strand. So we have our helix type thing where there's two strands together and they twist together. DNA is a double strand. Right? Because RNA is just one strand, it's much smaller. And this is why RNA is much smaller, so it can move in and out of the nucleus of the cell into the cytoplasm, so it can actually do the work in building proteins, actually reading the instruction manual that DNA gives it and building the different proteins that we have. So it's smaller. RNA is smaller because it has to move around. All right, so there's the big difference. We talked about most of those already. And so I already mentioned this here, RNA single strand, but we can still make a copy. So the big idea here is that we can take a strand of DNA and instead of making a copy of that DNA, instead we can just make a single strand of RNA instead from the DNA template. But the big idea is that instead of using T, we use U instead to complement our A nucleotide. And then in DNA, we use T. All right, but that was before. Now we're talking about RNA. These are the workers. These are the workers that are doing the building. Okay. That's the base pairing. G and C is still the same. All right. So just the U is different. So RNA, these are the workers that are actually going to build the proteins in our bodies. They're the ones that are reading the instruction manuals. DNA houses the instruction manual, but RNA actually reads it and does the work. So there's 3 different types of RNA, and we'll talk about mostly 1 and 2. So we have mRNA, rRNA, and tRNA. Thankfully that those beginning letters tells us a little bit about what it does. So mRNA is the messenger RNA. This is the RNA that is involved in transcription of DNA. So it's doing the copying. Okay, mRNA is doing the copying of DNA. And all of this is happening in the nucleus of the cell. I should mention this. I have a slide about where all of this is happening. And then all of this happens, the 2 and 3 happens in the cytoplasm. All right, so mRNA is responsible for the transcription of DNA. It's making the copy of DNA in the nucleus. So because you can't build proteins in the nucleus, it has to go outside of there into the cytoplasm to actually do the work. So messenger RNA is just making a photocopy of the instruction manual and bringing it outside so it can get dirty and do the actual work. The second type of RNA, rRNA, which is ribosomal RNA, is just a component of the ribosome. This is where protein synthesis happens. All right, it is another type of RNA. We usually give it cartoon looks to it to show, hey, this is where the protein is being built. So basically, rRNA is where your messenger and your transfer RNA kind of come together and work together to build your IKEA cabinet. All right, then the third one here, transfer RNA, this is the one that translates and reads the mRNA, the photocopy of the instruction manual. But the important thing with the transfer RNA, what it's transferring is it's actually bringing the correct amino acids to build the protein strand of whatever the instruction manual is saying. So transfer RNA here is bringing in the correct amino acids to build the thing that we want, right? mRNA, that's making the photocopy. tRNA is bringing in the correct materials to actually do it, right? So there we go. Got all that down? Got all that down? Okay, let's keep going. All right, so, all right, the first one, messenger RNA. So mRNA, remember, it's a single strand. It's making the photocopy of DNA. So here you can see, and this is all done by RNA polymerase, so an enzyme. Essentially, it's untwisting. Untwisting DNA and slowly making a copy, a complementary copy of your DNA strand. And again, remember, when we bring two strands together, they're always anti-periplanar, so there's a certain direction that transcription occurs. And remember, if you think about DNA as going from 5 to 3, if that's your DNA, I'm going to do it the other way. If your DNA strand is going from like 3 to 5, that means the mRNA strand that you get when you write it down, when you write the complementary code, it will go from 5 to 3. And that's kind of what you see here. What's being, this is the purple is your DNA strand, all right? Yeah, the purple, see how like right there? The purple, that is your DNA strand. And the blue one here, that is your mRNA strand. So note here, for DNA, it's going from 3 to 5. And what's coming here on the mRNA strand is 5 to 3, that we're building towards the 3 side. So what I like to call these 3 to 5 DNAs, I call that my template DNA, because the resulting mRNA will be in the correct 5 to 3 order. That's typically how we read it. So do note that the kind of the order direction of transcription really matters here. So yeah, this whole idea of getting that complementary base pairing is called transcription. We're making a photocopy of the DNA. Right? So we can bring it to the ribosome and the transfer RNA to do the actual building. I like to think of it as like if we think of like really important books in our world, like the Bible, the Quran, the Torah, all those things, it's like you would never read the original one. The original. We always read copies of it, right? And it's the whole idea here, like you would never touch, I guess, like the original one from I don't know, okay? You never touch the original one, but you would be able to, put in your pocket, put in your backpack, a copy of it, all right? And it's the same idea here, that you never touch the original one. You don't want to touch the original DNA strand, but so instead you make a copy with it so you can play with it. Outside of the nucleus in the cytoplasm. All right, so yeah, the big idea here, DNA template, mRNA are anti-periplanar. So I like to think DNA is typically the template is 3 to 5, mRNA, then you write 5 to 3. Okay, and then the rules still apply within there. Okay, So next, that's messenger RNA. It's making the copy. Ribosomal RNA, it's the components of the ribosome, so the site where the actual protein synthesis is happening. Note that I'm not actually showing any RNA strands here. Typically, ribosomes and ribosomal RNA is shown as these kind of pictures here, these little like funny hands. It's just a place for the mRNA and tRNA to come together and actually build the protein that we want. So in your biology class, you learned that a ribosome is one of the organelles with inside the cell, and this is the purpose of it. It's just a place for your M and tRNA to come together. There's two different units, okay? There's a small subunit and a large subunit, and what happens here is that mRNA comes out of the nucleus of the cell into the cytoplasm onto your ribosome. And so here is your strand of mRNA, and note that the nitrogenous bases are pointing up. All right, so they're kind of like flailed out. So what happens here is that tRNA can come in and read those nitrogenous bases that are sticking up. So your tRNA will go towards the big subunit and basically read the letters, the nitrogenous base codes that are being presented from the mRNA. So this is just a place where those things come together. All right, so that's rRNA. All right. Okay, and so then the last piece of RNA here is transfer RNA, tRNA. Sometimes they call this the clover shape RNA. I think this is a question on your homework. That's the only reason I bring this up. It's the clover shape RNA, because if you actually draw out what a tRNA looks like, it looks like this, okay? That this is a kind of the folding that you would see in transfer RNA, and each of these little ticks, like you see like on a zipper, each of those is a certain nitrogenous base, all right? So just know that tRNA is known as the clover leaf shape RNA because of when you kind of splay it out like this, it looks like a clover. In the book, though, they show transfer RNA looking like this. So this picture of transfer RNA is a little bit more representative of what it looks like in the body versus the clover leaf shape. That's what it would look like if it was flat. But tRNA isn't flat. Most things in our bodies aren't flat. So this is a better representation of what's going on. All right, so tRNA involved in the translation. I always forget whether it's transcription or translation first. I just memorized that it's in alphabetical order, okay, and that you have to copy the DNA first before you can read the copy. So it goes transcription first and then translation. Okay, that's how I memorize it. I don't know how you do it, but that's one way to do it. So tRNA does the translation. It's reading the photocopy that we made, which is mRNA. It's reading the mRNA that we made. So how it does this is what's called an anticodon on the transfer RNA. And it goes to the correct sequence on the mRNA looking for the complementary codon. All right, so what is a codon? It's a 3-letter sequence of nitrogenous bases. The question is, why 3 letters? Why do we have to have 3 nitrogenous bases in a row to determine what amino acid to bring in? It all has to do with statistics. Okay, all right, so the big idea here, here in this picture, you see everything together. The ribosome, the rRNA, the mRNA splayed up so tRNA can come in and read it. And then here is the tRNA, this yellow squiggle here, with its anticodon, with the complementary kind of code, reading your mRNA. And the big idea here with tRNA is that amino acid on the transfer RNA is covalently bound to that transfer RNA because it's transferring the correct amino acid to make the sequence. So the whole point of DNA, or this idea of RNA, and copying DNA, our instruction manual, is to make the proteins that make up our bodies. proteins are made up of units of amino acids. So the transfer RNA is reading the code to figure out what order of amino acids to place in your polypeptide sequence. All right, so let's look into this whole idea of the anticodon and codon and where that comes from and how to read them. All right, so putting it all together, let's actually read the genetic code. So I already mentioned that when we talk about codons or anticodons, we are talking about a 3-letter code. When I say letter, I mean nitrogenous base letter. I mean like A, U, G, or C. Okay, that's what I mean by letter. Okay, so we need 3 different letters to determine what amino acid to bring in. Do we remember how many amino acids there are? 20 in our bodies, and there's 22 within like all living things. But let's just say 20. So we need to think about statistically, how many letters do we need? How many different letters do we need in a row to make 20 unique combinations to basically indicate which amino acid to bring in? It's like the whole idea of like how many letters are in our alphabet? 26. How many words are there in English? Like a billion, right? So you can think about, we have 26 letters in our alphabet. We can arrange them in all of these different unique ways to make different words. So many words that I don't even know how many words there are in the English language. But for proteins, we only need 20 words. We only need 20 amino acids to call for. So if we think about it, if we have a 1-letter code, and so instead of having 26 letters in our alphabet, let's pretend we only have 4 letters in our alphabet. So if I'm trying to make one word, one letter, and I only have 4 possible like keys on my keyboard, but I can only place 1 letter, how many different combinations can I do? If I have a keyboard and there's only 4 letters on the keyboard, and I can only press 1 letter, how many different combinations, unique combinations can I do? 4, yeah, this is I'm tricking you, this is statistics, okay? Yes, I can only do 4 because it's only 1 letter. I can either pick A, I can pick U, G, or C, that's it. But then the question is, what if I have 2 letters? If I can put 2 letters back to back, the number of combinations increases. It's actually the number of letters, so 4 to the exponent n, so it's 4 squared. Because the first letter, I can pick A, U, G, or C. Then the second letter, I can pick A, U, G, and C, again, in any combination. So there are 16 unique combinations if I'm allowed to use 2 letters in a row. Does that make sense? Okay. So like baby statistics. All right, but that's only 16 unique combinations. That doesn't take care of all 20 amino acids I need. So I actually need a 3rd letter. All right, so instead, the different combinations that we have, it's going to be 4 to the 3rd instead, because I have 3 different possible letters that I can place in a row, and 3 different letters I can place in a row with 4 different letters to choose from each space. And so instead, it's 4 to the 3rd here. There's actually 64 combinations, unique combinations I can have. So 4 to the 2, 4 to the 1. So that's how we get statistically how many unique combinations we can have. So there are 64 combinations of letters A, U, G, and C within mRNA when we are looking at 3-letter codes. So those 3-letter codes on mRNA is called a codon. mRNA. This allows us to call up any amino acid that we want. It actually can call for a lot of different things. So the anticodon, this is on tRNA, and it's just the complementary 3-letter code on the other side. Complementary meaning that, we're using the rules A equals U, G equals C. All that stuff. So yeah, codon, I have it right there. The 3-base sequence in mRNA. So this allows us, because we have 64 unique combinations, we can call for any amino acid we want. There's also other kind of directions that an mRNA can give, and that's the stop codon and the start codon, and those have very specific orders of nitrogenous bases that you see here. So the start signal typically is when you see AUG, August, on a mRNA strand. AUG, when we look at the codon on the next, the genetic code on the next slide, it also is methionine. So oftentimes when you're building a protein, it'll start with methionine, and then at the end, once the protein's done, once it gets to the stop codon, it'll just cut off that methionine at the beginning, oftentimes. And then here are our stop codons. All right, so let's see if we can't do some, so that's the genetic code, okay? So the whole idea, we need 3 letters in a row, 3 letters in a row. So like A, U, G. How we read this genetic code is typically when we read a mRNA strand, we're going from the 5 end to the 3 end. So you kind of just chunk it in 3-letter pieces. And how I typically do is I start in the middle, see how there's a 5. And you look at that first letter, A, so I go over here, A. And then you look at the second letter, U, so then you go here. And then the last letter, the third letter, G, so you go here, methionine. And so this is start. This is how the mRNA, the tRNA knows where to start. It finds the AUG, August, start codon, all right? And then you just keep going. You just keep going. Have you seen this before? People know, yes? Okay. Let's do some practice, okay? You will always be given this. You would never have to memorize it, okay? Just, you need to know how to use it. So you start on the inside, you work your way out, given these different letters, okay? So the interesting thing here is because there's 64 combinations, see all of the different amino acids on the outside, that some of them double up because there's more options than there are unique amino acids. So like glycine, there's a bunch of different combinations that all mean glycine, right? So do we have an example? No, we don't. I just wanted to kind of put it together. We can do this example right here. So basically, putting it all together, if we are given a DNA template, remember, we can't read the book, the original book. We can't read it. We're not allowed to. It's too special. So we have to make a photocopy of it, of our DNA template with mRNA. All right? So this is the whole idea of transcription. This is happening inside the nucleus of the cell. So this is what this is representing. So do note here, look at the DNA template using our rules where A equals U, G equals C. We can make the mRNA, so pretend that that's not here. Let's just do it ourselves. Okay. When we want, a question can say, here is the DNA template. What is the corresponding mRNA strand? So the first thing that you should do is recognize, okay, if I make the mRNA strand, it's antiperiplanar. So I start with the 5 side, but I still go through the letters, okay? So T is DNA, right? So this would be A. So I should put this as well, all right? So T, since it's in mRNA, we use U instead, T would be an A. So then if we keep going, A would be U, C would be G, right? Yeah, and then you just keep going, making sure that instead of, if you were to write a T, you use a U instead. So C, C, so that's G, G, A is U, T, that is A, C, G, A, U. Is that right? Yes. And then the last one, GCA here, G would go with C, C goes with G, A goes with U. All right, and then we work all our way to the end to our 3 end. So that would be the corresponding mRNA strand. This is what's being read by your tRNA. And so note here that mRNA is smaller than the double helix of our DNA. So that mRNA can leave the nucleus and go to the cytoplasm where the ribosome is. Okay, that's the meetup spot where the mRNA and tRNA build the cabinet together. Where now the tRNA can come in, okay, tRNA, read the codon, so the 3-letter symbols here, to bring in the appropriate amino acids. So let's take our code here and see what tRNA would bring up. So I'm going to just copy this here. Let's go back to the codon. okay? And see if we can't read it. So AUG, we already did this one. So we start in the middle and we work our way out. So A, then we go to U, and then within that U, we go to G, and we see that that first three-letter code is methionine. It also means start. Okay. Then the next one, GGU. So we start there, like at that 10 o'clock. So G, G, and then U. So that's glycine. Do we all see how to read this? There's other ones that are more square looking. I don't like I like this one. This is the one that you will receive on the exam. If there's a question about, it will be in the question. It'll be in the question. Yes, so this is the one you will receive. Okay, all right, what's next? So the next three-letter code, AGU. So AGU, so that would be serine. All right, and then the last three-letter code is CGU. So CGU, arginine. Okay, so that's how you read the genetic code. So this is what the tRNA will bring in. It knows which amino acid to bring in by the anticodon, by the complementary three-letter code that goes with the codon, three-letter code off of mRNA. Yes? Yeah, so you have to transcribe first. Yes, so we cannot, like if, this is a good question, and we can actually do an example on our practice, whatever thing. There's a couple ways I can ask you this question. I would say, okay, what is the amino acid sequence? But there's two ways I can ask you. I can give you this strand. I can give you the mRNA strand. That's easy. You just chunk it up in thirds. You look at the chart. But what if I give you the DNA template strand instead? And I ask you, what is the amino acid sequence? So if I give you this instead, you can't plug this straight into the genetic code. You can't do it because it's the wrong way. It's the 3 to the 5. So the first step here, if I give you the DNA template, is that you need to make the corresponding mRNA strand, then read the code. Does that answer your question? Yes. So if I give you this, you can't go straight there. You have to convert to mRNA first. Okay, but if I give you mRNA, then yeah, go hog wild. Yeah. I will explicitly always say, this is the DNA template, and you will see 3 to 5. Or I can say, here is the mRNA code, and it'll be 5 to 3. I think let's just finish up and we probably have time for the example problem I have in the thing. All right? Okay, so here's all of it together. All right, so this is the same slide basically. So here's your nucleus. This is where your mRNA is making the photocopy with DNA. Since mRNA is small, it can leave the nucleus. It arrives at the ribosome with the rRNA, so the transfer RNA can come in, actually read the code, and bring in the appropriate amino acids. So here you see your growing protein strand, all right, to build whatever the cell wants. Sounds good? Okay, so those are the 3 different components. Okay. And then these last kind of things here basically just says everything that I just said. Okay. That protein synthesis starts with the mRNA, goes to the ribosome, transfer RNA reads it, and it's starting to translate your mRNA to bring in the correct amino acids. And then here's just some notes about termination and when to stop. Essentially, the tRNA will find the stop codon, which you see on the genetic code, and that's when it knows when to stop. And when the stop codon is reached, the polypeptide will break away from that from your rRNA, from your ribosome. And then the note here, since the start codon is typically associated with methionine, that is also removed from your peptide chain. Okay, so that is nucleic acids. I think I didn't have enough practice problems here. So why don't we just bring up, okay, let's bring up let's go here, or maybe not here, go to the Oh my gosh. Come on. Where's my stuff? All right, whatever. The resources for the final page. Okay, go there. And download it's called the in-class/Zoom packet, all right? It's a bunch of questions. It's like 13 pages of questions. We will work through this today on Tuesday and, on Thursday and for the Zoom thing. Okay, so let's download this, and I'm gonna direct you to like the last page, the page 12. Create new note, whatever, go. Okay, let's go to page 12 and see if we can't kind of put it all together. Recent notes here, nope. Where did I just? Okay, here. All right, so let's go to page 12. And see if we can't do some of these questions, kind of wrapping up chapter 11. And the key's already posted here, so if you want to work through it on yourself, that you can do that. Let's actually start on question 43. Okay. Let's consider GMP. What components are in GMP? So GMP describes what type of thing? We just got done talking about nucleic acids. So GMP is a nucleic acid. So the letters kind of tell you what's in it. So if you recall, nucleic acids have three components: a nitrogenous base, a sugar, and a phosphate. So here, since it says MP, MP stands for what? Monophosphate. So you know for a fact it has monophosphate in it, or a phosphate. So here we have G stands for which nitrogenous base? Guanine, okay? It has a sugar with it, but which one? Ribose. Yes, it has ribose. What would it be if it was deoxyribose? What would the symbol? What would the letters be instead? DGMP. Yes. So this is ribose, and then MP means it has a phosphate. Yeah. So these are the components. Okay. Yes. So then, is GMP? Is it a nucleotide or a nucleoside? Tide, because phosphate group means that it's a tide. If it's just the first two, if it's just guanine and the ribose, it would be a nucleoside. Right, finally, GMP would be found in what? DNA, RNA, or both? How do we know that? How do we know it's only found in RNA? The ribose, okay? So I want you to be like looking at the right places within here. Okay, all right, so here we go. Sequence of bases in a DNA template. This is the DNA template. What is the correct sequence in the corresponding mRNA strand? So here's that sequence. So whenever we make a complementary strand, it is always anti-periplanar, meaning that that 5, the corresponding strand will always start at 3. Okay, but then we still go through the letters the same way. But this is mRNA, so we need to make sure that we're using U's instead of T's within here. If I could change this question to what is the complementary DNA strand, and you would just use T's as normal. But since this is mRNA, we use like this way. All right, so U goes with, so I always like to just remind myself A equals U, and sometimes T when it's DNA, and then G goes with C. So U is analogous to A, G is C, U is A. Sometimes I like to break it up. Oh, there's only 2, that's all right. There's only 2 letters at the end, so there's not groups of 3. We're just coming up with the sequence. We're not doing the amino acid strand here. ACA, A goes with U, C goes with G, G goes with C, and the next one, UA, U goes with A goes with U. All right, and then that last is 5. So sorry, that last one, this is what happens when I write my own questions. It doesn't have a third one on that last one, but that's okay, because we're not actually determining the amino acid strand. The next question, this would be the correct sequence, and that's fine. So do make sure that you have the right number of letters to match whatever your template strand is. If I had that third one, there would be another within there. So the one thing to note here, pretend I did have like an extra letter or whatever, like there were sets of 3. we could not write the amino acid sequence from this because it's the wrong way. We would have to flip it because we always need to read it 5 to 3. So do make sure that you're reading the correct order here of your nucleic acids. So let's go to the next one where we actually are given an mRNA sequence. And so do note here I give you the genetic code. I will always give you this genetic code, the wheel within here. So here, this is, I'm telling you, is mRNA. So you don't have to go and change, find the complementary strand. You can just start with this strand and group it in groups of 3. So here, good, I gave it in groups of 3. So we just read it as is. UGU, I like doing these. Do you like doing these? Yeah? UGU, so U, G, U, that first one is cysteine. So then A, C, G, threonine. And the last one, UCA, so UCA, so serine. You got that? Yeah. Don't start with what? Methionine. Well, that's only if the first one is AUG. Yeah, so I just happened to not make it start with AUG. Okay. You start it with meth if AUG was there. So let's do one more. How about, what if I gave you the DNA template? I'm just going to make stuff up. It might not even make sense. What if I gave you the DNA template of 3? So I'm starting with 3. Let's say, okay, I'm just going to do it backwards. ACU, GCA, UGU, 5. So let's go through the strategy here to get your amino acid sequence. If the question says, here's your DNA template, what is the amino acid sequence that's produced? Can you just plug that into the genetic code? No. You have to make the mRNA sequence first. So this is DNA. Oh, wait, no, I have U's in it. I can't do it. See, I'm not a biologist. Sorry, let me just change it real quick. There we go. Okay, change all those U's to C's. Okay, there we go. So let's try this again. So here's your DNA template. What is your amino acid sequence that's produced from tRNA? So the thing to note here is that I give you this DNA template in the 3 to 5 order, because when you write your mRNA strand, it will be in the 5 to 3 order like we typically read it. So that's typically how questions are asked here. If I give you a DNA sequence and I ask you to kind of do your mRNA and then get amino acid, I have to give you it in the opposite direction here. So we'll start with the 5 side, so it's the same idea. A goes with U or T, and then G goes with C. So in this case, A is U, G, A, C, G, C, A is C, G, U, T, G, T is A, C, A. Is that what other people got? Yeah? Okay, good. All right, so now this is what we plug into the genetic code. I don't know what amino acids this produces. Does it not work? Ah, dang it, the first one's a stop. But you get it, right? You get it. All right. Okay, it's all right. I just made it up. I made it up in my head. So yes, if you actually take this code, yes, what you get is like stop ones. The first one's a stop. But you just report whatever actual amino acid is there. That's what happens when I just make one up in the top of my head. All right, so on Thursday, we will continue with this packet. Okay, sound good? C, G, U. ACA. Yes?
Should ones for 44 BTs also instead of U's?
Yeah, it should. This is what happens when I write my own things.
Do we have to memorize what people
No.